Okay, welcome everyone. Thank you for attending some informational talks on DILIsym services. My name is Liesl Schoda and I'll be moderating this session. I'm a principal scientist and director of immunology at DILIsym services. Today, it's my pleasure to introduce Dr. Zachary Kenz, who is scientist 2 with DILIsym services. Dr. Kenz received his BA in mathematics and computer science from Concordia College, and then also was awarded a master's and PhD from North Carolina State. He worked at Brunel University as a research professor, and then was also on the staff at MIT Lincoln Laboratory prior to joining DILIsym services, where he has done extensive work in developing mathematical QSP models of various injury or disease conditions, as well as applying those to address questions in drug development. So with that, I would like to turn the presentation over to Dr. Kenz to introduce us to DILIsym services.
All right, thanks, Liesl. So I'll be giving kind of an overview of, DILIsym services, sort of, where we started, which is sort of modeling drug-induced liver injury, and then, as the title kind of implies, beyond that. So, the quantitative systems pharmacology, systems biology modeling world, and where we've started and where that's led us as a company to extend our modeling efforts within safety, within liver injury, and then some really exciting areas that we've been working to build out our modeling efforts. So to kind of dig into that, we'll begin with an introduction to DILIsym services as a company, and then get into a bit about QSP modeling, kind of our philosophy, but also how that ties into kind of the broader QSP field and philosophy. And then I'll discuss a little bit about some of the platforms that we've been working on and developing over time for almost a decade now, actually, in terms of DILIsym, and then some of the other platforms over the past few years, and then kind of wrap up at the end, and also allowing some time for questions. So in terms of DILIsym services as a company, originally, we'll talk a little bit about this, DILIsym started as an initiative focused on liver injury, and was a company kind of developed focused on that, and then has had a fruitful sort of development partnership, and then ultimately was acquired by Simulations Plus. And so part of that Simulations Plus family develops modeling and simulation software in pharmaceutical, biotechnical industries, and there's a wide wealth of knowledge and information that we've developed. So we'll talk today and focus more on the QSP side, but throughout Simulations Plus, there's various development efforts all the way from prediction from structure to pharmacokinetics, pharmacodynamics, to QSP, to working with data. And so there's kind of a nice synergy between the different the divisions within Simulations Plus to really bring forward modeling and simulation prediction within pharma. And so that's from drug discovery to clinical development research, regulatory submissions, and we kind of develop different portions of that with different software platforms that help different aspects of pharma that can work independently but also together. And so like I've alluded to, DILIsym in particular focuses on quantitative systems pharmacology, systems biology models, and quantitative systems toxicology to support efforts in the pharmaceutical industry to address questions of toxicity and safety as well as efficacy. And so to that end, we began kind of in the safety toxicology side and have been building out different platforms to support the development of safe and effective drug therapies. So really the overall goal is understanding biology, physiology, understanding drug interactions with that biology and physiology to answer key questions that the industry might have as drugs are progressed through the pipeline. So to do this, to sort of bring this effort to bear, you might imagine that this takes a team. So we can't just rely on one particular type of expertise. We actually need to bring together people from many different backgrounds. And so we have a really a nice wide array of backgrounds, a growing team of people. And that begins with some of the efforts of Paul Watkins in the upper left, our initiative founder, scientific advisory board chair, Scott Seiler, Brett Howell, our chief scientific officer and president, who bring different backgrounds in scientific fields. Sean O'Connor, the CEO of Simulations Plus, who has a wide array of industry experience and technical experience. And then our principal scientist, Elise Lashota, who you just heard speak, who's focused in who focuses in immunology, but also clearly has, along with Sergey, clearly have Sergey Ermakov, clearly have a wide array of knowledge. And then, our entire team varies knowledge in biology and engineering. So we have different, some people who have a more like chemical or biological engineering background, mathematical backgrounds, and then we as well have software engineers like Corey Berry in the middle, who all together can bring these pieces of development to build out these biological models. And as I've alluded to, the team consists of different people, different backgrounds, and we need these aspects on multiple projects. So when we build a team, either to work on a consulting project with a company or to build out and develop a platform like DILIsym or some of the other platforms I'll discuss, we need this mix of engineers and life scientists that can work closely together, both on the model development and construction side, but also the validation side, to take into account the available data that we might find in the field, different ways of constructing models to capture the physiological properties and constraints that we see, and then to actually make sure that we're answering the key questions that we see in the field. So, when we think about constructing these models, we want to keep in mind a good balance of representing the physiology accurately, but also making sure that we're not sort of digging too down into a rabbit hole. And that's where having this mix of people is really nice. As you can imagine with a team, while we're growing, it's still kind of a small team, so everybody wears multiple hats. So we have our kind of major development projects, but we also contribute to various aspects of running a division, this kind of a smaller division of a larger company. And this environment, so obviously I'm kind of sitting here presenting in my remote workspace, and so I'm one of the full-time remote workers. Other members of our team are full-time remote. Some are on site in our base in North Carolina. And so when we kind of look at other skills in addition to the technical skills that are required, we do need to effectively communicate. Within our team, when we're working on development projects, we'll have different remote meetings, kind of like this go-to session, but also we have a lot of remote meetings with clients. And so building out those communication skills is a key and critical part of the development, but also the ultimately the deliverable portion where we can inform and communicate with different industry partners to make sure that we're meeting the safety and efficacy needs of the field. So I've alluded to the fact that that DILIsym in particular has been around for a while and been in development for a while, and the initiative that the DILIsym, the company, is continuing to shepherd was built on a public-private partnership. And so because companies are concerned about safety, there's a very interesting relationship where there's some desire and willingness to share some safety information. And so this DILIsym initiative was created to bring together knowledge, data, information from different pharma companies under one kind of umbrella, so that safety information, toxicology information on the liver side could be brought under one whole and assessed. And then each of those members would then have access to that information as an initiative member. And also, since the members would have access to that sort of safety information, they could guide areas for model development. And that's how over time there have been phases to an initiative, and that examples right now we're working on adaptive immunity, and that's something that's been guided by interests of the initiative over time as the models continue to develop and build out from the initial stages of just kind of getting base toxicity and base key sources within the model. And then ultimately the goal is to provide insight into mechanisms of toxicity, but also as the field develops, we want to have the model be able to incorporate novel biomarkers. So as data comes out, we can incorporate the mechanisms and the ideas in the field into the model. In addition, over time, the FDA has become an active contributor to efforts. They've supported 2 ORISE fellows, and also we've had a good relationship in terms of the FDA obtaining licenses and also coming to some of our training, so that there's a knowledge base when DILIsym models are submitted or communicated, that the FDA can assess what's under the what's going on in terms of the DILIsym modeling. So the overall goals are always go back to improving patient safety through these sort of modeling efforts. Ultimately, we want to help drug development efforts so that safer drugs make it out to people to help people. At the same time, because we can do more testing in silico, in simulations, a long run goal is always that we hope modeling can reduce the need for animal testing. And then also because simulations, you don't have, maybe you could potentially reduce clinical trials, you could focus clinical trials. The idea would also be to reduce cost and time necessary to develop new drugs. So some of these have certainly borne fruit over the course of the time since the initiative was started in 2011. Various pharma companies have seen a benefit to this. 19 companies have participated at different points in time. And then what's been incorporated into the software as a result of this partnership are compounds data and experiments that have added to the knowledge base within DILIsym. And we're continuing to develop this core initiative to support DILIsym development. So that's where the QST and core initiative started, and where all the systems modeling that underlies liver biology got DILIsym, began the basis of DILIsym. From there, we've begun branching out into other areas. So if we understand liver biology well, we could branch into something like non-alcoholic fatty liver disease, or NAFLD-sim. And then that's more on the efficacy side. And then from there, you might say, okay, we understand toxicity, so we could go from liver to kidney, and that's RENA-sim. And if we understand NAFLD, that's a fibrotic disease, so we could start looking at other fibrosis in other organs, IPF-sim. If we understand QSP modeling, we could go to other areas like radiation. And so this is how some of our expertise and understanding of how to work with data and build really high quality quantitative systems pharmacology, toxicology models has expanded into meeting this vision of safer, effective, more affordable medicines for patients through modeling and simulation, and expanded into these different platforms that are shown in the icons in the middle. And at this point in time, DILIsym and NAFLD-sim are available for licensing, training, and development, and also consulting projects. And then our RADASym radiation platform, RENA-sim, and IPF-sim software are in development. Again, ultimately with the goal that those would be available for licensing and training and development. And also, as part of this, we need data to input into the model. And so for DILIsym in particular, but also DSSI, you know, we look at other aspects. So data interpretation, and then in vitro assay experimental design and management. So sort of looking at the whole picture of managing data and managing the sort of model inputs, design, and assessment of model outputs. And, you know, as part of that, working within model development at DILIsym, we've distributed time between consortium and proprietary projects. So that initiative fits under the consortium projects where we are directing based off of members' input each year. We're incorporating new drugs into the software. We're looking at various aspects of modeling, which would be adding new pieces of the physiology that could help improve the predictive nature of the software. So I already mentioned the adaptive immune system. We've added other submodels recently, some that have to do with some of the bile acid submodel or related to that model, so that we're kind of expanding aspects of DILIsym. And then also we incorporate variability into the model, and there are simulated populations that we are considering and working to add to assess different population variability. And then also, in terms of adding to the drug pipeline, you are interested in kind of early development, from very early development to preclinical, like mouse, rat type of testing, the animal testing. And so we're you know, always looking at adding additional species there. We have mouse, rat, and dog in DILIsym, so we're always looking at adding more species there to expand the capabilities and the assessment of abilities. And then there's ongoing efforts to look at software speed and performance. So in addition to biology, there's always software associated and kind of wider population type assessment that goes along with building a platform. And then in terms of proprietary projects, we've worked with various pharmaceutical companies, big and small, and for DILIsym, we're looking at hepatotoxicity risk. So if their compound is showing liver signals. And the idea there is, since DILIsym is a systems pharmacology model, we're able to look at the actual mechanisms that might be at play in assessing the risk of liver injury. And so sometimes that drives new development, sometimes that helps them assess whether their compound is able to move forward in the pipeline. So that's kind of where DILIsym has been, and what the core of, you know, where we've looked at modeling efforts. In terms of the QSP modeling perspective we have, you know, I think I've kind of alluded to a lot of these, but we're designed to characterize biological systems representing disease pathophysiology, drug pharmacology, and pathways of toxicity. So that's a canonical sort of description of QSP models, or as close as you can get in kind of the QSP world. And the key is that we want to represent different aspects of the physiology and then interactions between drugs and these biological systems. So we want to see how much drug, in the case of DILIsym, is getting into the liver, and then what it's doing to the liver, particularly if it's causing injury. And then this can identify data gaps and then potentially guide future experiments. So there are technical skills required, modeling, parameter estimation optimization, coding, reasoning, and then things like network biology and physiology. So some of these aspects are philosophy. Some of these are kind of in the wider QSP world, which is why we have a white paper cited here. And so the idea is to, again, build systems models that you can then look at changes to parts of that model and see how it affects downstream, say, biomarkers or measurable markers that you can compare to data. And the idea is to answer key questions. So you look at those downstream markers, and then you want to answer key questions about what mechanisms might have changed, and then see those effects in the physiology. And so I've hopefully mentioned mechanistic effects a few times, and that's where some of these models might differ or might build out some of those mechanistic effects more so than other types of modeling, which focus on different aspects, like pharmacokinetics, pharmacometrics, machine learning, or artificial intelligence methods. Some of those are more, they have mechanistic, kind of a scale of mechanistic modeling built into them, depending on where they might sit. But they often tend to be based a bit more in the statistical world, and we're looking at addressing different key questions. So QSP is really focused on mechanistic insight. So it takes a little while to develop the models, but when you've got those models built, the idea is to look and say, okay, this is, within the body system, this is exactly why this change is occurring, and to be able to then build out different hypotheses and do hypothesis testing within the mechanisms that might be at play. We talked about updating and expanding models to fit available data and needs of clients. And then in terms of like the actual language that we're using to describe the interactions, we are working with ordinary differential equations, ODEs. That's provided in a kind of an appropriate mathematical language, balance of description and speed. We certainly, would expand to other types of mathematical equations if needed, but it's a good way to combine interactions with sparse data. And we're working in a MATLAB environment currently, but we can expand and are expanding to other languages, looking at Julia as a potential language for development, but also, simulation speed, maybe production at some point. And then our main software platforms are being converted to C++ for client use. In terms of model development, we're always researching new topics, looking at new modeling for models in terms of the underlying equations, and then looking at predicting novel therapeutic safety and efficacy. So some of that development is guided by consortium members, some of it's guided by clients that looking at new diseases or organs. We are certainly keeping up with the scientific literature that will inform potential model updates. And then, so that's like our internal efforts. And then we also are looking at attending conferences like various like Society of Mathematical Biology, ACOP, other places that allow us to present seminars, talks, and posters, and then publish in journals and ways to interact with the wider field to make sure that there's vetting of our models, but also feedback in terms of the wider community giving direction to where we need to take our development. In terms of the platforms that we're looking at, we've seen these DILIsym, NAFLDsym, RENAsym along the top row. So DILIsym, there's some development motivated by the consortium, but also there's some FDA interaction because there is this safety need. So the FDA does kind of put requirements on companies in terms of what they need to show safety-wise, and the goal was to assess liver toxicity. NAFLDsym, there was development motivated by corporate sponsors, and there are some key liver fat, inflammation, and fibrosis aspects to that model to assess efficacy. RENAsym is kidney toxicity motivated and was sponsored by a small business grant. And then the bottom row, IPF-Sim, there was a corporate sponsor that was motivating that development, and that's focused on lung fibrosis and function loss. And then the RADAsym radiation model that was also sponsored and focusing on the effects of radiation. And also the idea was to help garner approval for treatment of compounds under the animal rule. So there we're trying to use preclinical species where you can do some radiation experiments that you wouldn't be able to do in humans to predict the effect of a compound in humans. And that's where the animal rule comes in. And then MitoSym was developed to generate parameter inputs from assays, in part to understand those, but also as input into other platforms like DILIsym. So these are our platforms. Again, we've built out some of them and then are continuing to work on them. And then in terms of like our development over time, when you're building models, and part of the reason that we get into this as a company and want to build all this out is that model development in one platform impacts development in another platform. And so I wanted to just quickly kind of look at how that's worked within our platforms that I just showed. So DILIsym is an acute injury, short time frame. Usually, drugs are dosed, maybe once or a few times over the course of a week, and so you get injury quickly. And within the innate immune system, there are macrophages and neutrophils that participate in the immune response. So there's initial injury, and then there's an innate immune response as well to some of the damage. So that sort of model was changed, certainly, to reflect the fact that then in DILIsym, that's a chronic injury platform. So it's a longer time frame. The injury signals are different. The magnitude of injury is different, but the core equations and the core cell sort of responses, I mean, a macrophage is still a macrophage, is just responding to a different signal. We could take some of that knowledge and bring it from DILIsym to DILIsym. Well, then we could take some of that knowledge from neutrophils and macrophages in DILIsym and bring it to IPF-Sim. But at the time, because that was a platform where we needed to describe some of those interactions a little bit differently, we were actually able to split out portions of the macrophage structure a little differently. So we had some of this incorporated, but there are macrophages that reside in a tissue, and then there are macrophages that are recruited from circulation in response to injury. And we did a little bit of restructuring when we were developing IPF-Sim. So then we could take that structure, ultimately, we'll bring that back in a second, in the acute injury phase. And so for DILIsym, we brought in macrophages and neutrophils, but also needed dendritic cells, so we used some of the same structure. And then we took this new macrophage structure that we had developed in IPF-Sim and brought it into DILIsym. So you're seeing this like acute injury phase, a chronic injury phase, these models developing different signals, different aspects, but ultimately we're taking knowledge and learning from one platform and using it to inform some of the structural and regulatory aspects in other platforms. And what's interesting about this is it's all gone full circle in that this new macrophage structure we're now incorporating into DILIsym. So we're bringing all this knowledge kind of back to the original platform. And because we have this knowledge about life cycles and life cycle regulation, even though radiation is like a completely different platform, it's not macrophage, neutrophil, dendritic cell based, we could use some of the structural formatting from these platforms for bone marrow resident cells. And so there's a lot of knowledge that you can take from different platforms as you're developing other platforms, even though the regulation and the cells might be different. And we're continuing to refine these over time. So you may refine because of data, but you also may refine because your cell structure just looked different in a different platform, and you can take that mathematical structure and learning and bring it into one of your existing platforms. And I would imagine we'll ultimately kind of complete the cycle back to DILIsym at some point. So that's, again, part of the biology, part of the modeling and development. That's really interesting when you're developing a QSP model over time. And then across the company, we have different platforms. So our DILIsym, IPF-Sim, DILIsym, and DILIsym sit in the preclinical and clinical space. But in terms of the overall company, we have various platforms that sit in the predictive space in terms of where dosed compounds can get into cells and assessing some of that. So if there's interest in looking at all the way from discovery through preclinical to clinical, we can talk about some of that as well on the side. So ultimately, DILIsym, there's a really engaging sort of dynamic environment within the team and as well within the field. And there's an opportunity within the company to contribute to these different models, expanding upon existing models and developing new models, and then ultimately benefiting the pharmaceutical industry in a number of different ways in terms of predicting safety and efficacy for different therapeutics as well as working ultimately from predicting from structure, which one of the case studies that Christina will go through in a second will use some of that predicted from structure. And within companies, this potential for mathematical modeling has really kind of come to the forefront in different groups. And within regulatory agencies is becoming more recognized to expedite these processes and improve drug development and also efficiency. So that's kind of the summary. And then if there are questions, but also we have our various LinkedIn accounts that you can grab a shot of here to see sort of and connect with us there. So if there are questions, I can take those. Otherwise, I can let Christina go into the case studies as well. After Lisa, I think, takes some time here.
Thanks, Zach. That was a great intro to DILIsym and some of the work that's gone beyond. For the audience, if there are any questions, please enter them in the chat. In the interest of time, we'll move on to the next presentation, so to put all the questions and commentary discussion at the end of Dr. Batista's presentation.